This is section 2 over mitosis. So why your cells might want to do mitosis? Basically to grow more cells. So when you grow taller, you grow more bone, more skin tissue, you're always going to have to create more cells, and so mitosis does that. Some organisms use it as a form of asexual reproduction. Little invertebrates called hydra will just bud off. Basically, a part of their body falls off and it becomes a new one. And that is the same basic principle as mitosis. And to repair damaged tissue. Let's say you fall off your bike and skin your knee, there's some tissue missing, and the cells will divide to try to fill in that tissue. So mitosis is basically when the cell's nucleus divides. So obviously this only happens in eukaryotes because prokaryotes don't have the nucleus. The cells are going to divide into two daughter cells, and those cells are going to be exact replicas of the original cell. So if there's 46 chromosomes in your original cell, each of the new daughter cells are going to have 46 chromosomes in each. And this is done in somatic cells. And a somatic cell is all of the cells in your body except for the egg and sperm in your body. So your skin, your nerve, your heart muscle, every cell in your body is going to be doing this. Okay, some terms that you're going to need to know. A chromosome is basically DNA that has been coiled up and is short and fat, and then they're going to be linked to their sister chromatid. Now, if you remember, in the cell cycle that we just learned about, the sister chromatids are made during the S phase of the cell cycle. So, at this point, when mitosis is getting ready to start, every cell has a copy of each chromosome in the cell. So, original human cells have 46 chromosomes. When mitosis starts, when M phase starts, there are actually 92 chromosomes at the beginning because each has a sister chromatid. A gene is the little segments of these chromosomes that actually codes for a protein. And these are essential code for the things in your body that you really need, your proteins. Stages of mitosis go prophase, metaphase, anaphase, telophase, and cytokinesis is the last part of telophase. The rest of the life of a cell is in interphase. So remember, interphase consists of G1S and G2 of the cell cycle. Okay, in prophase, basically what's going to happen, the chromatin that is in your nucleus is going to have to coil up. If you remember from our cell coloring picture last unit, we had chromatin look like a bunch of spaghetti noodles. It's too unorganized like that. So basically what they're going to do is they're going to wind up and curl up until they become short and fat, and that's when they're called chromosomes. And they're going to be joined with their sister chromatid at a place called the centromere. So here you can see these black dots are the centromere. Each of these is an identical copy sister chromatid. Okay, in addition, the centrioles are going to start making a spindle, which is made up of microtubules, and then the nuclear envelope is going to dissolve, so now the chromosomes are free floating in the cell, and the spindle is going to start attaching to these little centromeres, and that's what we call the kinetochore. So this spindle is going to have to organize itself so it's attached to every single one of those centromeres. Okay, so here you can see an actual cell during prophase. This is going to be an animal cell, circular. You can see that the chromosomes are kind of visible. If you were to look at a cell in interphase, this would all be a jumbled mess. But now you can actually see individual chromosomes, but they're still not organized. So that's prophase. Metaphase. All those chromosomes are going to line up along the middle, which we call the metaphase plate. The centrioles get positioned at the north and south pole of the cell. Okay, all of these spindles are attached to the little centromeres in the middle. And basically the whole point of this phase is to line everybody up. Okay, and then the sister chromatids are going to be on each side of the plate, and then the spindle is going to start shortening and start pulling them away from each other. So metaphase, here's a plant cell in metaphase, they're all lining up in the middle. Anaphase, those spindles are going to reel in just like a fishing line. It's going to get shorter, and it's going to pull the chromatids away from each other. So anaphase is for away, and the poles are moving farther apart. Notice this is not a circle anymore, it's more like an egg shape. So the cell is becoming elongated. Okay, So the spindle is getting shorter and shorter, pulling those chromosomes away from each other. So now all of these chromosomes are the same ones that were on the other side. They're exact copies. Okay, telophase is when the nucleus is going to come back. Basically, it's the opposite of prophase. So the nucleus comes back, the chromosomes start to lengthen back out into chromatin, and the spindle dissolves and disappears. You're then going to get a little pinching off in between called the cleavage furrow, and that's basically a new cell membrane being formed. There's a checkpoint here to make sure everything is okay in each of the correct cells before it is actually totally separated. And cytokinesis is when the cell is dividing everything else in its inside into the two new cells. Because not only does the cell need a whole set of DNA, but it also needs some ER, Golgi's, mitochondria, everything. So it has to divide up all of that cytoplasm into the two new cells. Okay, in an animal cell, cytokinesis just looks like that pinching that we saw, making a new cell membrane. In a plant, because they don't really have that loose cell membrane, they're going to form a cell plate in the middle, which is a new cell wall. Okay, so here you can see that cell plate dividing in the middle. Okay, so just a review, this is happening during the M phase of the cell cycle. And we have prophase, when everything gets organized and gets shorter. The chromosomes become shorter, the spindle forms, nucleus goes away. Metaphase means middle. They're lining up along the equator. Anaphase, they're pulling away from each other. And telophase, they are separating into two new cells, and the nucleus is coming back, the spindle goes away and dissolves. I'm going to post a couple extra links on my website if you would like to explore any of these reviews, just in case you didn't understand anything.